Let's be honest, modern biology is absolutely drowning in data. The amount of information we're getting from genomics, proteomics, and all the other omics has created a massive challenge. It's not just about the experiments anymore. It's about how we store, share, and, most importantly, make sense of this digital flood. So today, we're going to decode the fundamental language of bioinformatics, the file formats that make it all work. Okay, just look at this graph. It shows the explosion of digital information in the world. And biology's story is basically this on steroids. You've got public databases like GenBank that have been doubling in size every 18 months or so. Think about it. The Human Genome Project, ENCODE, the 1000 Genomes Project, these massive efforts are churning out petabytes of data. We're talking about the genomic blueprints for tens of thousands of organisms. This, right here, is what big data in biology actually looks like. And yeah, big data comes with some pretty big challenges. It's not just about finding enough hard drive space to store it all. How do you even begin to analyze billions of DNA sequences? How do you curate all that information to make sure it's accurate and actually meaningful? How do you search it, share it with a collaborator across the planet, and visualize it in a way that sparks a new discovery? Honestly, each one of these bullet points is a huge field of research in its own right. This slide just perfectly captures the core problem we're facing, using a couple of old parables. You've probably heard of the blind man and the elephant. You know, each scientist might be studying one tiny piece of the data, a single gene, one protein, a specific pathway, and they might describe their piece perfectly. But they're all at risk of completely missing the bigger picture of what the whole elephant looks like. Or think about Plato's cave. Are we just looking at the data, which are like shadows on the wall, without understanding the true biological reality that's casting them? Without some kind of common framework, we're all just guessing. So, how do we see the whole elephant? Well, the solution is actually pretty simple and elegant: standardization. By agreeing on a common set of file formats, we create a shared language. It's like a lingua franca for bioinformatics. It's what allows a researcher in Tokyo to make sense of data generated in Toronto using tools that were built in London. This is the invisible infrastructure that modern computational biology is built on. All right, let's start with the absolute basics, the formats we use to represent the linear sequences of DNA, RNA, and proteins. You know, the alphabet of life itself. The most stripped-down format is called appropriately raw, and it's exactly what it sounds like. It's just a string of characters, like something you'd type into a plain text file. There's no title, no description, zero context. It is pure, unadulterated data, and nothing else. And right away, you can see the massive problem with this. I mean, look at these sequences. You have absolutely no idea what they are. Okay, the top one is probably DNA, the bottom one protein, but from what organism? What gene is it? Are they even related? You just can't tell. This format is really only useful for tiny, specific scripts where you, the programmer, already know all the context. So to fix that glaring problem, we got the FASTA format. This is, without a doubt, one of the most common and important formats in all of bioinformatics. And it solves the context problem with one simple, brilliant rule. The file has to start with a single description line, and that line must begin with a greater than symbol. That's it. The sequence itself follows on the next lines. And here's a perfect example. That greater than symbol is the magic key. Everything on that first line, the gene name, the species, that's the header, the metadata. Everything below it is the sequence. This simple structure made it so easy for programs to read that it just became the universal workhorse for the entire field. Now, if FASTA is like a quick ID card, then the GenBank format is the full, detailed biography. This format was created because as research scaled up, just knowing the name of a sequence wasn't enough. Scientists needed a way to computationally link all kinds of biological features directly to that sequence. And this is the standard that major public databases like NCI-BI use to capture all of that rich context. Okay, let's break down a GenBank file. It's split into different sections. The header, which you see here, gives you all the critical background info. Locus is the unique name. Accession is that stable, permanent ID that you'd cite in a research paper. Definition gives you a quick summary. An organism tells you the full taxonomic lineage. It even has references to the scientific papers where the sequence was first described. But really, the true power of the GenBank format is in this part, the features section. This is where biological meaning gets mapped directly onto the sequence coordinates. So you can see an entry here for CDS, which stands for coding sequence. It tells you the exact region that gets translated into a protein. It even provides the translated amino acid sequence right there in the file. This is what lets you connect the gene to its final product, and it's what makes this format so incredibly valuable.
And then finally, after all of that rich annotation, you get down to the origin section. This is where the actual sequence itself lives, often with numbers on the side for easy reference. The whole record then ends with 2 forward slashes, which is just a signal to any program that this entry is officially done.
So, the big takeaway here is that you've got to choose the right format for the job. RAW is for quick and dirty scripts. FASTA is the universal currency that nearly every analysis tool will accept. And GenBank? That's the archival format for deep, detailed analysis and for submitting your data to public databases. It's always a trade-off between simplicity and how much information you need. Okay, so far, everything we've talked about has been a one-dimensional string of letters. But we all know biology doesn't happen in 1D. It happens in 3-dimensional space. So now we need to look at the format we use to describe the actual physical shape of molecules.
For 3D structural data, the standard is the PDB, or Protein Data Bank format. These aren't just sequences anymore. These text files store the precise spatial coordinates of every single atom in a molecule. This is what allows us to build and visualize a 3D model on a computer.
The heart of a PDB file is the atom record. Each one of these lines represents a single atom in 3D space. It tells you the atom number, what kind of atom it is, like a carbon alpha or an oxygen, which amino acid it belongs to, and finally, its exact X, Y, and Z coordinates. A single file can have thousands and thousands of these lines. And when you plot them all, you reconstruct the molecule's beautiful, intricate 3D shape. This is the kind of atomic-level detail that makes things like drug design and protein engineering possible.
All right, let's shift gears now and talk about the formats that were born out of the modern sequencing revolution. We're talking about high-throughput machines that can generate millions or even billions of sequence reads in a single run. The ABI format actually comes from the older Sanger sequencing method, but it's super important because it captures more than just the final string of A's, T's, C's, and G's. It's a binary file that stores the raw trace data from the machine, the chromatogram that shows the fluorescent signal for each nucleotide, and that is absolutely essential for quality control. And this is what that trace data looks like. You see the 4 colored peaks? Each one represents one of the 4 DNA bases. A nice, clean, sharp peak means the sequencer was very confident in its call. But if you see messy, overlapping, or low peaks, that's a red flag. It signals that there might be an error or some ambiguity, and it lets a human go back and visually inspect the quality of the raw data. For modern next-gen sequencing, the undisputed kings are SAM and its binary cousin, BAM. A SAM file, that sequence alignment map, is a text file, so you can actually open it up and read it. And what's so important is that it doesn't just contain the millions of short sequence reads, it also contains all the information about how and where each read aligns to a reference genome. The BAM file is just the compressed binary version of SAM, so while you might look at a SAM file to troubleshoot, all the heavy-duty computational analysis happens on the BAM file because it's way smaller and dramatically faster for computers to work with. And it's worth quickly mentioning the SFF, or Standard Flogram format. This one is a bit more specialized. It's a binary format that was used by pyrosequencing platforms like 454. These machines worked a bit differently. They detected flashes of light as nucleotides were added, so the SFF file stores that raw flogram data. It's just another way of capturing the machine's raw output.
So, when you step back and look at all of this, from that simple little greater than symbol in a FASTA file to the mind-boggling complexity of a BAM file, what you're really seeing is the shared language that powers the entire global project of computational biology. This is what makes discovery at scale even possible. It's what allows data to flow from a lab to a consortium to a supercomputer and back again. But that brings us to a huge and I think really exciting final question. The formats we've talked about are great for describing static, linear sequences and structures. But biology isn't static, right? It's dynamic. So as we move deeper into an era of single-cell multiomics, where we can measure the genome, the transcriptome, and the proteome of one cell all at the same time, and as we start using AI to model these incredibly complex systems, what will the next generation of data formats even need to look like? What will that next common language of biology be? That is the grand challenge for the next decade.